to discuss the development and progression of the disease known as Huntington's disease, which is proposed to be a prion disease. To really understand Huntington's disease, I need to do a brief recap on the central dogma of molecular biology. Here we have DNA in the nucleus, and hopefully we know that DNA can be transcribed into RNA, and that mRNA can be translated by a ribosome into a protein, a process which occurs in the cytoplasm. Also, the DNA is going to have to replicate whenever cells divide. And so whenever a cell divides, it has to double the amount of DNA that it has so that it can portion one of each chromosome into each of the resulting daughter cells. And so we have this replication cycle right here. So how is it that replication of DNA can lead to Huntington's disease? Well, to answer that question, we need to think about the gene encoding the protein responsible for Huntington's disease. The protein itself is called Huntington, as you can see written right here, and the gene itself within the DNA has a repeat pattern of GTC, okay, and it has a bunch of these. And really, depending on the individual, there's a different number of repeats of these three nucleotides. In general, what we normally say is that if you have less than 36 of these repeats, here's 36 right here, 1, 2, and then 34. If you have less than 36, you will probably not have Huntington's disease. There seems to be a critical threshold when you get to 36 or more of these repeats of GTC that you end up getting Huntington's disease. Now, GTC is a nucleotide triplet in DNA. When it's transcribed into RNA, that is mRNA, you have a repeat of CAG, and then this repeats the exact number of times that you had in the DNA. Now, the CAG codon, when this gets translated into a protein, that is the Huntington protein, which is abbreviated HTT, CAG as a codon encodes the amino acid glutamine. And so if you, for example, had 36 repeats of GTC, then your RNA is going to have 36 repeats of CAG, and your protein, somewhere in the structure, will have a repeat of 36 glutamines, okay? So if you have seemingly a random arrangement of nucleotides of A's, T's, C's, and G's, it's not really that difficult for the polymerase to add the bases without much error, okay? But if you now put in these repeats, okay, GTC, 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 and it's having to replicate this over and over again, what can happen is when the polymerase is moving over this, the polymerase can actually forget which of these triplets it was on, and it may inadvertently add in an extra GTC or a couple of them. And this is actually why individuals have varying numbers of GTC repeats within the Huntington gene. And so what replication actually does whenever the polymerase makes an error is it can actually add in extra GTCs. To conceptualize this, imagine saying the letters ABC over and over and over and over again, and try to keep track of which ABC you're on. So ABC, that's 1, ABC, that's 2. So you just go ABC, 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 and you do this 20 times, there's no way you're going to remember which ABC you're on. It's the same for the polymerase. So the polymerase can forget, essentially, in quotes, which GTC it's on, and it can inadvertently add extra GTCs. And this is the mechanism by which individuals get more and more of these GTC repeats, therefore more glutamine repeats. And they're not exactly sure why this is, but whenever the resulting protein from the Huntington mRNA has 36 or more glutamines, it tends to make that protein susceptible to misfolding. And if you go back and watch the introductory video for this playlist, we talked about how misfolding proteins is really the major mechanism behind prion diseases. Okay, so when this Huntington protein misfolds, possibly due to this repeat pattern of glutamine, of 36 of them, it tends to form the prions. And remember, with prions, those proteins tend to have a higher degree of beta strands. That's a secondary structure, and it compromises the amount of alpha helices. So when you have the switch from alpha helices to beta strands, it tends to cause these misfolded proteins to aggregate together. And so when Huntington forms its prion form with the more beta strands, it'll form Huntington oligomers. So very similar to the way when you have fold-up chairs, if you fold those chairs to where you store them, you can stack a bunch of those fold-up chairs on top of each other. That's kind of what Huntington will do. It'll kind of stack with itself, and it'll form initially oligomers, which will eventually lead to large aggregates. And these aggregates of, in this case, Huntington, are generally what are thought to propagate really any prion disease. In the case of Huntington's disease, the neurons that have a tendency to be affected most strongly are going to be cells, or I should say nuclei of the basal nuclei, which some sources still call the basal ganglia. This is a cluster of cell bodies within the brain that are really responsible for the control of movement. So that's what leads to the loss of movement control and inability to repress unwanted movements, and then given time, it will actually lead to the death of the individual, unfortunately. And what we'll see over the next few videos is that many of these prion diseases are going to operate by extremely similar mechanisms. Now, the protein of interest is going to be different in each case. For example, in Parkinson's disease, the protein is not Huntington, it's actually a protein called Parkin. In the case of Alzheimer's, it's thought to be a protein called tau and also beta amyloid. There's actually 2 proposed ones in that case. But the general mechanism is going to be very, very similar.